This is my DNA model. So the first thing I'm going to talk about is the bases. The bases are thymine, adenine, cytosine, and guanine. So these are the information in the DNA that determines what the proteins are and what the amino acids are, and pretty much what we are. And so then there's the backbone, and so the backbone is made up of a sugar, which is deoxyribose, and a phosphate. And so this backbone holds together the whole DNA. And so the sugar always goes with the base, and the phosphate always is in between the sugars and the bases. And so they're right here. This is sugar, this is phosphate, this is sugar, this is phosphate. And so in reality, a DNA is in a double helix structure, so it's like this, where it's kind of like twisted. And so this is how all DNA is actually coded, and this is how all DNA actually is. And so I'm going to try to explain the mutation in this. So over here, you can see over here you can zoom in. This is a thymine and an adenine over here. But if for some reason the adenine gets taken out, and is replaced with a cytosine, this one is replaced with cytosine, then we get a mutation. So then if the body messes up, and so when the polymerase comes and checks the DNA and it messes up, and then it takes out and sees the thymine doesn't match with the cytosine and the adenine, because adenine is supposed to match with thymine and guanine is supposed to match with cytosine. So this is the correct pairing. But when it sees the incorrect pairing, then it will try to correct it. So there's a possibility that the body will take out the thymine and replace it with a guanine. And so it could potentially be like this. And so this mutation is called substitution. It just changes one base pair, but that could actually change the whole amino acid and the whole protein. And so the next thing I'm going to explain is the RNA. So the RNA is pretty much so the DNA will unzip during transcription. It will unzip, and then the polymerase, which is an enzyme, will come over, and then it will look at this, it will read this, and then it will make a copy of it. So let's just say the polymerase comes, and so it comes and it checks through all of these, and so it unzips this, and then it makes a copy of this. And so it looks at this and then makes a copy, because the base pairs have to align. So then they'll align it like this. It'll read this and it'll make a copy based on the alignment. So pretty much we have this is called the RNA. This is ribose nucleic acid. And this is very similar to DNA. You still have the 4 base pairs, but so you have adenine, guanine, and cytosine, but instead of thymine, you have uracil. So this is just different than thymine. It kind of replaces thymine. And so then the backbone is also phosphate, sugar, phosphate, sugar. And so 3 of these is called a nucleotide, if I haven't mentioned that earlier. This is a nucleotide. And so the backbone, this is actually one-sided. This is called messenger RNA. Okay? And so it's still a backbone of phosphate, deoxyribose, phosphate, deoxyribose. And so this is a copy. But, okay, so let's just assume earlier that it was back to the original. It was thymine, adenine, thymine. So the mutation did not occur. And so when it copied it, it would copy an adenine. But if there was a mutation, it would copy a cytosine. This is very important. So let's just assume there was a mutation. And so this piece of RNA, it leaves the DNA, it leaves the nucleus, and then it enters into the ribosome. This ribosome, it will read the RNA, it will read each chunk, and then every three bases, it will create an amino acid based on the 3 bases. So this is called translation. And so for my example, I have 3 of these, and 3 of these. So then it reads it like this, and then you get this. So then I kind of drew this model off, but it's supposed to be like this. So the first one is lysine, and the second one is leucine, and the third one is valine. And so it's just like this. So this one, this AAG, this is lysine. The middle one is leucine, so CUC, and then the GUA, this is valine. And so pretty much these are amino acids, and a lot of amino acids make up a protein. So we can just say this strand of messenger RNA created this protein, okay? But if we don't have the mutation, or if we have the mutation, so let's just assume this cytosine is gone, and then we're gonna put in an adenine. Right, so if we put in the adenine, we could get a completely different amino acid. So right here, you can see it. Instead, you have lysine, asparagine, and valine. So this is different. And so the middle becomes from lysine to asparagine, and the other parts don't change, but this middle part could change, which means it could alter the DNA by a lot. And so this mutation could actually change how the person ends up. Okay? And so let me try to explain the proteins more closely. So we're just gonna say this is a protein, okay? And so this has a unique structure. It's made up of 3 amino acids. And so then I've labeled these amino acids, and like shown earlier, they match with the messenger RNA. And so this is the original. This is the mutation. You can see there's a little bit of difference in the middle. The leucine becomes the asparagine. And so then maybe this trait could code for, let's just say this could code for rough thumb. So your thumb is rough. And then this one could code for smooth thumb. And so if there was a mutation, someone's thumb could become from rough to smooth. Okay? And so I'm going to try to explain transcription again. So pretty much transcription, I'm just going to sum all this up. Transcription is when this DNA unzips, and then it codes. It looks at the opposite side. So in this thing, thymine, cytosine. The opposite is adenine, guanine. And so it's going to unzip, and then it's going to make an RNA copy of this. Adenine, guanine. So like this. But then they're going to notice this mutation, and they're going to try to correct it, okay? This is pretty much what's happening in transcription. It's making the copy of the messenger RNA. Translation is when this goes into the ribosome. It goes into the ribosome, and it reads it, and then it forms 3 of these form 1 amino acid. So if we do this, then AAG forms lysine, AUC forms asparagine, and GUA forms valine, okay? So this is transcription and translation summed up. And so also it was hurt by the mutation like I mentioned earlier, so thank you.